Good morning, teacher. Good morning, students. My group and I are going to present about clonation. Here's our structure for our project. We have introduction, general focus of the problem, objectives, theoretical framework, and conclusions and recommendations by bibliography, students, and the last part, the systems.
The introduction. Want background, a student needs to learn the importance of cloning as future means of reproduction in species. 2. Research environment, working in school in our English hour, investigate and at the same times question about cloning and its advantage and advantage. General focus of the problems. 1. Limits of the research. Working on cloning as a subject of investigation, analyzing Dolly as a cloned mammal, discussing advantage and disadvantage. S2. Statement of the problem. Cloning as future for species.
Objectives. General objectives, students will understand that animal cloning gives a reproductory opportunity and at the same time the change to preserve endangered species. A specific group objective, understand importance of the cloning as means of the reproduction in species and the cloning of Dolly the sheep as the example. Theoretical framework. What is the cloning? Cloning can be said to be identical copies of the organism's cell or molecular. There are three different types of cloning. Genetic cloning creates copies of genes and segments of DNA. Reproductive cloning creates copies of whole animals, and therapeutic cloning creates embryonic stem cells.
Disadvantage of cloning. Abnormalities seen in cloned animals included weight gain, malformation, reduced viability at partition, respiratory problems, fetal livers, dilatation, epidermal hemorrhagias, and kidney abnormalities.
Cloning can be very bad for the humans or animals. There are also cloning that are specially made to harm the species or person.
Cloning can have disadvantages such as the malformation of the cloned animal organisms, and also the cloned subjects may get off the hand and be dangerous, since this method is not yet at its exact point to be safe.
I believe that cloning is something totally dangerous because it affects the animal that is being cloned, such as its health, and this can cause the death of the animal. Also, cloning in general is very dangerous since it can produce serious diseases that could cause death.
Cloning can lead to negative downsides. It has not yet reached its best point if an animal is cloned. The animal will die due to the low defenses it has or even due to genetic mutations that it may have.
Yeah, a disadvantage of cloning in animals could be gain bad formations, reduce viability at birth, respiration problems, delayed liver of the fetus, epidermal hemorrhages, and kidney abnormalities in addition to animal abuse.
Conclusions and recommendations. Conclusion. Cloning is a kinetic method that is not effective since the animal is not possible to replicate or clone any species identical to the original one. But so complex human beings, animals, and plants cannot be perfectly copied. Recommendation. It's recommended not to clone living beings since they are not possible because the bodies are very complex and that is why they cannot be cloned to exactly to the original.
Come to the conclusion that there are clones that can be effective for both humans and animals, as there are also conclusions that can be harmful to some species. Recommendation: I recommend that each cloning they do is for the good of people or animal, because if they only clone for experimentation, they can harm the life of several species.
My conclusion is that cloning can bring problems in if something that bad is cloned, since it could alter the genes of a species and that it is not the same or that it does this in a few days.
I, I came to the conclusion that cloning can become very dangerous since it deserves risk as if the anomaly or living organism that you clone has not control and that is very dangerous for everyone.
Here we can see some devices used in clone research.
Here we can see some images about cloning, and the conclusion of our group is that if the cloning method is not used well, it can be very risk for us and another living beings.
Here's our bibliography of our investigation, and this is the project. Thanks.